Hello everyone, welcome to this short 15-minute video on blood glucose regulation. This is a topic that is on the Aspects to Human Physiology unit. It will be probably on the end of year test. If you're not doing the end of year test, then it is not on the alternative assessment in terms of the essay, but definitely worth knowing about. So what we're going to do is explain how blood glucose is regulated in relation to homeostasis, and then we're going to talk about the cause, treatment and symptoms of type 1 and type 2 diabetics. So you should know the difference between the two by the end of this video if you didn't know already. You probably know that one is genetic, one is lifestyle orientated, but I'm going to put a bit more science behind it. Chances are in your professional career you're going to come up against people that suffer from an inability to maintain their blood glucose levels. So it's really important for you to know as a healthcare professional what's going on inside the body, what you're going to do to treat it and how that treatment works. So I hope you find it useful and enjoy. So blood glucose regulation forms part of homeostasis, which means that we are balancing an internal condition. This is known as the set point. And the set point, or the point at which our blood glucose is normally set at, is 4 to 5.9 millimoles per litre. Depends on what source you use, but 4 is the floor. If you remember that, that's great. 4 is the floor. So when I sort of teach this, I always recommend that learners use these 4 boxes to revise, to learn, and then once you've got the 4 boxes, you can then remember it much better under pressure, is what I found. The only other things I want to add on are these, which is a B and an A, and we're ready to go. So blood glucose is maintained by negative feedback, in which case it's SRIA, as we can see down this left hand side. Now the stimulus might be us eating a meal. So this set point here is called a preprandial set point, which means the set point at which our blood glucose should be before a meal. So when we eat a meal, our blood glucose goes up. Now at this point I have to emphasize that we are measuring blood glucose. It's the amount of sugar within our blood. It's not the amount of sugar that's in our body, within our cells, within our water within our body. It's the amount of the amount of sugar that is in our blood. Okay, so when you eat a meal, it goes into your mouth, down into your stomach, into your small intestine, and it's there that it gets absorbed. The sugar gets absorbed, and that's only when our blood sugar increases, which is this line down the left hand side. When our blood sugar increases, that is the stimulus. Now what detects that? What detects the rise in blood sugar? And that is the reason why I put this little B on top of the set point, because this stands for beta, and in fact beta cells of the pancreas. That is the receptor and indeed the integrator for blood sugar rise. Okay. So the beta cells of the pancreas are monitoring the blood sugar, and we've had a meal and our blood sugar has risen. So that responds by releasing the hormone that we probably all heard of, insulin, which has the response of reducing blood sugar back to normal. So stimulus, blood sugar goes up. Receptor and integrator is the pancreas, specifically the beta cells. The effector would be insulin, and the response would be that the blood sugar goes down. The question is, how does insulin work? Well, if you look down this bottom left hand side, here's a bit of a diagram. Here the blue circles represent a cell membrane. I'm not drawing the phospholipids. There's a protein channel, and here is a receptor protein. This is the inside of the cell, this is the outside of the cell, and here we have a blood vessel that's running just outside of this cell. In this blood vessel, yes, we've got lots of erythrocytes, red blood cells, we've now got insulin, which are these black squares that I'm drawing, and nice colour, we've got these blue dots here, which are glucose. So when this insulin travels around the body, these black squares, they're slowly released out of the bloodstream and they bind to receptors on the cell membranes of nearly every cell in the human body. This joining of the receptor causes this channel to open. And remember, how does glucose get inside a cell? From the cell biology unit, you should be screaming facilitated diffusion. We go from a high concentration to a low concentration through a protein channel. And there's an extra, why do they do that? Well, for energy. That's why glucose has been, which is entering the cell. So by doing that, by opening these channels, you're taking the glucose out of the blood and putting it in the cell, which has the effect of, the response of, lowering blood sugar, and that's how blood sugar level is maintained. However, sometimes we pig out. Sometimes in this Sunday roast we eat far too much glucose than our cells are actually using or need at that particular time. So it makes sense that, and it's also evolutionary, that we've, if we've worked really hard hunting and we've killed an animal, for example, and we're having a feast, but we don't want to waste that opportunity. We've spent a lot of energy, a lot of time gathering and harnessing our food. So it makes sense evolutionary wise that we've come up with a way of storing that energy to be used at a later date. So if we eat too much sugar, then what actually happens is that we store it as something called glycogen, and that's usually in the liver or the muscles. And to know about glycogen is really, you should have done it a little bit in your macromolecules, but glucose is a monosaccharide. So if I draw these dots, these black circles here, they're all individual sugars and they're called glucose. And they're in the blood at the moment. If you send this glucose to the liver, for example, and you chain these glucose molecules together, you get glycogen. So glycogen is a polymer of glucose. It means it's just a long chain of glucose. Now the fact that you've taken this glucose out of the blood, delivered it to the liver and chained it together as something different, i.e. glycogen, you actually reduce blood sugar back to the set point, or close to the set point. And that's one way in which we deal with high blood sugar. This process of creating glycogen from glucose is called glycogenesis. Genesis meaning creation in the sort of Christian biblical sense. So glycogenesis, which can be seen in the word world to the side if you can't read my handwriting. So that's glycogenesis. Now, what happens if we're short on sugar? We've missed a meal, or we're starving, we're in the desert, we're trying to survive, whatever. Then our blood sugar goes low. There's my pen. And that would be another stimulus. Our blood sugar goes below this 4 level. The floor is the floor. Well, a different cell this time, and that is an A, if you can believe it. And it's an alpha cell. Again, of the pancreas. But this time, the alpha cells detect low blood sugar, and they release a hormone called glucagon. Not glycogen, glucagon. It's a hormone. Put H for hormone. H for hormone. And so what that hormone does is it travels. Try and follow my spotted line. This hormone travels to the liver, or the muscles, to my knowledge, and causes a reaction where it splits up this stored glycogen. So it splits it back up into its individual glucose molecules and dumps it back in the blood, with the effect of rising blood sugar back to normal. So basically it's raiding the larder and putting it back into our bellies, or in this case into our blood, and that's how we deal with low blood sugar. That process there is called glycogenolysis. Lysis means splitting. We've come across that a few times already. Lysis means splitting, so it's the glycogen splitting as opposed to glycogen production, which is genesis. And that is how we maintain our blood sugar levels. Now we've got two problems, and you'll come across these before. We've got diabetes mellitus, type 1 and type 2. Now the symptoms are the same, but the cause and the treatment are different, because the cause is different, if you see what I mean. So type 1 is genetic. I'll put genes because I can't write genetic on the end. It's an autoimmune condition, and therefore the white blood cells actually destroy the beta cells of the pancreas. Nasty. So if these genes turn on, they destroy the beta cells of the pancreas, then A, they can't detect a high rise in blood sugar, and B, they can't release insulin. If they can't release insulin, blood sugar cannot go down because the receptor proteins aren't stimulated and the glucose channels stay shut. So the blood sugar will stay high. Why is that dangerous? Well, we need the glucose for energy. If the cell is not getting the glucose, it can't create the energy as well, and therefore that's why it causes some symptoms such as dizziness, lethargy, leading to coma, cell death, that kind of thing, which is horrible to think about. Another symptom is that this blood becomes very viscous. It becomes very thick and sticky. So when the heart is trying to pump this around the system of capillaries and blood vessels, it's very hard. The heart has to work harder. So there's an increased chance of sort of heart disease and also stroke, because it won't fit through narrowed passages very well. If this capillary suddenly becomes blocked and it's very thick and sticky blood, there's probably more of a chance that it's going to get blocked and not get through and deliver the oxygen and glucose that it needs to for the rest of the body. So that's type 1. Type 2 is lifestyle. It's caused by lifestyle. So people eating too much sugar, people not exercising enough. And what happens is that these proteins on our cell membrane down here, don't remember about protein synthesis. It's a process that's continually happening. It will respond to the environment. So if you're bombarding it with sugar and sugar and sugar all the time, the cell will just become immune to that. They'll become immune to the signal pathway in which your cell is triggered to take in glucose. So essentially these receptor proteins become redundant, and they actually get, I think, taken off the cell membrane, so they become less sensitive, or the cell becomes less sensitive. And so no receptor proteins equals no opening of channels. Now we're saying no, there's going to be some happening inside the body somewhere, but not enough. So if these channels stay closed, we get exactly the same symptoms that we did with the type 1 diabetes, the sticky blood, the low oxygen, the low energy, the lethargy, the coma, the cell death, that kind of thing. But the cause is that it's self-inflicted. The cells are becoming that used to sugar that they eventually turn themselves off, and the receptor proteins get taken off, and the insulin just doesn't work anymore. Now relatively recently, in fact, probably the time that I've been teaching Access, which is 10 years or so, in that time it's been proven that you can reverse type 2 diabetes, and so a really low calorie, high exercise diet can start the protein synthesis production again, and those receptor proteins being put on the cell membrane. So that's one way in which we can treat type 2 diabetes. In terms of type 1 diabetes, there is no sort of reversal like that. We can't just start turning on or turning off the white blood cells and destroying the beta cells. So what happens is that type 1 diabetics have an insulin shot. So whenever they have a meal, they can have an insulin shot, which does exactly the same thing as it should do. Opens the glucose channels, and the glucose will go into the cell for energy. So hopefully that covers everything. That's how our blood sugar is regulated. That covers the cause, the symptoms, and the treatment of diabetes type 1 and type 2.